Hello, welcome to Biotanime. Staining. Staining is an auxiliary technique used in microscopy to enhance contrast in the microscopic image. It will help us to explore the microscopic world around us. The staining can be categorized based on the types and number of dyes used. They are 1. Simple stain. In a simple staining technique, a positive stain is used since the bacterial cell wall and nucleic acid is negatively charged. Example: crystal violet, methylene blue, safranine. Simple stain is used to provide color to transparent bacterial cells. To determine cell size, the morphology of the bacteria, and bacterial arrangement. 2. Negative stain is used to demonstrate the capsule-producing bacterial cells, which do not take up any stain. In this technique, a drop of bacterial suspension is mixed with negative dyes like Indian ink or nigrosine. Since the surface of bacterial cells is negatively charged, they will repel the stain. Hence, the bacteria will appear bright on a dark background. This technique is used to view capsules containing microbes. 3. Impregnation methods. This method is used when the bacterial cells and structures are too thin to be seen under the light microscope. They are impregnated using silver salts on their surface to make them visible. For example, the demonstration of bacterial flagella and spirochetes. 4. Fungal staining. To stain fungus, the LPCB stain or lactophenol cotton blue stain is used. As the name suggests, the stain has lactic acid, phenol, and cotton blue in it. The lactic acid will preserve the fungal morphology and acts as a clearing agent. Phenol acts as a disinfectant. And cotton blue is the chromogen in this dye. It will stain the chitin present in the fungal wall. The dye also contains glycerol, which will prevent the cells from drying. This technique will help us to view the shape, size, and arrangement of spores and hyphae. 5. Differential staining. They are used to differentiate two bacterial strains present in the smear. Usually, two types of stains are used for this purpose. The commonly used differential staining techniques are 1. Gram staining. Based on the stain taken up by the bacteria, they are classified into gram-positive and negative. The two stains used in this method involve crystal violet as primary stain and safranine as secondary stain. After staining, the addition of decolorizing agent will cause the thick peptidoglycan layer present in the cell wall of gram-positive bacteria to hydrate and shrink, which causes the closing of pores present in the cell wall. That prevents the stain from exiting the cells on the washing step. Gram-negative cells, on the other hand, have a thin layer of peptidoglycan and a thick layer of lipid will lead to an increase in the pore size on the addition of decolorizing agent and will allow the stain to wash out in the next step, and will get stained by the secondary stain. 2. Acid-fast staining, also known as Ziehl-Neelson staining, is used to stain mycobacteria. It will distinguish bacteria as acid-fast and non-acid-fast bacterium. In this staining procedure, carbonyl fusion dye is used. It will solubilize the lipid present in the cell wall of mycobacteria. When the heat is applied to the mix, the carbonyl fusion will penetrate the cytoplasm. This will give the cell a red color. 3% HCl and 95% of alcohol is used as decolorizing agent. The mycobacteria is resistant to this decolorizing agent due to the presence of lipid in their cell wall. But the non-acid-fast bacteria will easily get decolorized. The counter-stain methylene blue will stain these decolorized non-acid-fast stains. 3. Endospore staining is used to stain and view the endospores in the bacterium. Endospores are highly resistant metabolically inactive structures formed during unfavorable conditions by bacteria. Hence, they won't take up any stains. To stain endospore, Schaefer-Fulton's method is used. In this method, the primary stain malachite green is forced into the endospore with the help of steaming. Since malachite green is water-soluble, it has a low affinity to the cells and will be washed away with water. The counter-stain safranine will then stain the vegetative cells.
Thank you. Please like, subscribe and share. See you again in a new topic.